Hello, everyone. I am Toshiaki Koike-Akino from Mitsubishi Electric Research Laboratories, Mall, Massachusetts, USA. Today, I'd like to talk about chemical polar coding for molecular communications. This shows the outline. First, I want to talk about molecular communications. Then, we introduce some DNA engineering technologies. Using chemical reaction network, CRN, we want to provide DNA polar codes. Let's look inside of our body. There are many molecules working for us. Those molecular biology is really amazing. Recent advancement of nanotechnology can manipulate some materials at an atomic to molecular scales. Then in future, we can consider nanomachines who can provide some simple functionality, sensing or actuations. For example, using DNA, we can store large amount of data size. Even we can engineer a bacteria to work for us. In far future, we may consider some nanomachine network to perform some complicated tasks. Those nanomachines may cooperate each other. But how to share data among nanomachines are a key problem. Molecular communications were proposed as a new paradigm, which may not rely on electromagnetic fields, but molecules. This may be biocompatible and probably no energy required. There are many applications, including advanced health monitoring, nanomedicines, environmental sensing, chip fabrications, food engineering, and tissue engineering for the Internet of Nanothings. Comparing to the traditional communications, molecular communications uses molecules, not electromagnetic waves. And nanomachines may use chemical reactions. We may need some error control schemes. In molecular communications, there are three types of channels: directional propagation, semi-directional propagations, and random walk propagations. The first directional propagation may use molecular motor. Semi-directional propagations may use chemotaxis. And the last random walk may be based on diffusion. Molecular motors are really interesting. For example, kinesin and dynein are inside our body. Those proteins walk through microtubes inside the cell. This shows very interesting videos. As you can see, kinesin proteins are like a robot having two legs, and it walks through microtubes. And it can carry a big cargo from one to another end. The second method using chemotaxis use bacteria to lead one end to the receiver end. The bacteria can carry plasmid from transmitter to receiver. The speed of the transmission is not that high. It's like 1 centimeter per hour. Another biological molecular communications are diffusion-based. For example, cell-to-cell communications are based on diffusion of molecules through gap junctions. Under diffusion channels, nanomachine may encode digital data 1-0 into a modulation signal, like molecular emission rates. Then receiver try to decode 0-1 data. There are some studies of theoretical analysis. For example, channel capacity was derived under the assumption of stochastic molecular biology. Not only theory, but also some prototyping and experiments were reported. There are some different digital modulation schemes for molecular communications. For example, emission rate, molecular type, and temperature change. But mostly, many molecules are required to send one bit. Another type of molecule are based on DNA. The DNA molecule can accommodate multiple bits at once. For example, a simple plasmid can store up to 32 kilobits. DNA are encoded by 4 types of nucleobases, A, G, C, and T. Amazingly, recent advancement of DNA engineering could realize a new DNA computers. It can solve traveling salesman problem or some gaming toy problems in experiments. In 2006, MIT developed a new DNA circuit programming language, Cero. This is based on Verilog HDL language. We can program any digital circuits into a DNA sequence. This shows an example of on-off circuits based on 3 proteins A, B, and C. The synthesized DNA can produce a sensor on and off depending on the existence of A, B, and C. They validated their method in experiments. Given DNA strands, many molecules can be generated, and those molecules can react each other, and very complicated behaviors can be established. And it is known that stochastic chemical reaction network is Turing universal machine. Therefore, we can perform almost any arbitrary computations using just molecules. For example, 2017, Columbia University showed an experiment of DNA fountain calls to transfer a tiny operating system data for DNA storage. This may realize 200 petabytes per gram. The error correction capability of fountain calls can protect the entire operating system data from the error. Our talk is a proof of concept to use polar calls. The polar calls were proposed by Aricon in 2008. It achieves capacity in any arbitrary channels. Coding and decoding are low complexity, and it was applied to the 5th generation standards. The polar code uses polarization phenomenon, which uses polar kernel when it takes 50% erasure probability. Upper branch provides worse erasure probability of 75%, and lower branch provides better channels having 25% erasure probability. And repeating polarizations, we can create very bad subchannel and very good subchannels. We can polarize the reliability. And the fraction of the good subchannels can approach the channel capacity. Aricon proposed successive cancellation decoding. First, gathering all the likelihood information to decode U0, then subtract U0 to decode U1. And cancelling subsequently, we can achieve capacity. By integrating cyclic redundancy check and risk decoding, we can outperform state-of-the-art low-density parity check calls. This figure shows the performance of bit error rate of state-of-the-art LDPC calls and also polar calls for the code lengths 256 to 16,000. For all the cases, polar calls can be better than LDPC calls. In particular, polar calls can be much better than LDPC at shorter code lengths. And more interestingly, the nonlinear complexity of polar calls can be a benefit compared to linear complexity LDPC calls. Although polar calls can be higher complex at a very long calls, shorter than 4,000 code lengths, the polar code can be even lower complex than LDPC. In this talk, we propose chemical reaction network for short polar code. To realize chemical polar codes, we may use DNA programming. Given DNA strands, we can establish chemical reaction networks. CRN is a programming language for chemical systems. It comprises a set of reactants, products, reactions, and rate constants. The DNA hybridization is a multi-step process. However, It can be modeled as a single bimolecular reaction, A plus B to C. Assuming that the system is well mixed, then the chemical concentration change can be governed by an ordinary differential equation. For realistic chemical reaction simulations, we use ChemPy. The bimolecular reaction A plus B to C can realize mean function. It is because production of C can stop at the minimum concentration of A or B, as shown in this figure. We consider some basic chemical operations for polar cores. The first one is bit representations. We use two molecules, A0 and A1, to represent A equals 0 or 1. In order to make A0 or A1 exist complementarily, we may use three reactions. The probability representation is just a ratio of the concentration of A0 versus A1. The multiplication over two probabilities, PA times PP, can be realized by three reactions. And the division of the probability can be established by two reactions. For polar encoding, we may use exclusive OR binary operations. Chemical exclusive OR can be realized by 8 reactions. The first 4 are producing C1, depending on A and B, and the last 4 are generating C0. For chemical successive cancellation decoding, we consider two types of operations at upper branch and lower branch of polar unit, F function and G function. Those functions propagate probability of A and B. The F function is based on sum of products. And in chemical reaction, we use multiplications and summation to realize F functions. The G function requires divisions as well as multiplications. After propagations of the chemical probability, we use hard decision at the end of the layer, depending on the concentration A0 and A1. This can be realized by 5 reactions. Let's validate our proposed chemical SC polar decoding. This is for polar cores of size 4. The CRN is formed by using ChemPy. As you can see this figure, the concentration can converge towards the ideal likelihood over time. For such a short cores, we can also consider maximum likelihood decoding. For ML decoding, we first calculate code word likelihoods. Then later, we calculate bitwise likelihoods, which can be realized by 8 reactions. This figure shows the simulated validation of ML decoding. We can observe that chemical ML decoder can converge faster than SC decoder. Another benefit of ML decoding is the size of chemical reaction networks for short code words. For example, 4-bit polar cores require more than 200 chemical reactions for SC decoder, while the ML decoder uses only 44 reactions. Of course, the complexity of ML decoding is exponential, and it does not scale up to the longer cores. Let's conclude my talk. We first introduced recent advancements of molecular communications and DNA engineering. We then proposed a new chemical reaction network for DNA polar cores. We developed parallel SC decoding and ML decoding. We confirmed that our chemical reaction network can converge to the idealistic equilibrium. We analyzed the required number of species and reactions. We found that ML polar decoding converges faster than SC decoding and much simpler at short code word lengths. We just showed the proof of concept of chemical polar cores, and more rigorous analysis in realistic in vivo channel models is left as a future work. More questions? Please contact me.